Alzheimer's disease. It is neurodegenerative disease caused by two misfolded proteins, which are produced by our bodies, not this dietary proteins. These two misfolded proteins are amyloid beta protein, tau protein. Let's have a look at the misfolding of amyloid beta protein. In the cell membrane of neurons, there is an amyloid precursor protein. It has three segments: N-terminal end, beta segment, C-terminal end. As we know, protein gets broken down after use, and the individual amino acids are stored in our body for further work. The amyloid precursor protein is broken down by enzymes such as beta secretase and gamma secretase. Beta secretase enzyme cleaves amyloid precursor protein at N-terminal end, whereas gamma secretase enzyme cleaves amyloid precursor protein at C-terminal end. The cleaved segments are released outside the neuronal membrane. The cleaved segments are in their pure form, hence they are insoluble. Amyloid beta segment in insoluble form is hazardous, because all the released monomers of amyloid precursor segment bond to each other at their sticky ends, and as a result, it causes accumulation of amyloid beta protein. This accumulation gives rise to the formation of insoluble fibrils of beta, pleated sheets. Microglia cells are responsible for the elimination of protein aggregates. These cells take up some beta-pleated sheets. After this, the microglia cell gets activated and releases inflammatory cytokines. The inflammatory cytokines causes neuronal damage. It might even become a hurdle in neuronal synapse and disrupt the signaling process. It can also get deposited around blood vessels and lead them to burst. Now let's have a look on misfolding of tau protein. Tau protein act as a support to the microtubules, so microtubule don't fall off. Microtubules acts as a railway track as they ship nutrients and molecules alongside. Accumulation of amyloid beta segment. Secretase kinase enzyme. The tau protein gets hyperphosphorylated by kinase enzyme and leaves the microtubule. This tau protein bonds to other hyperphosphorylated tau proteins and forms a neurofibrillary tangle, and as a result, the microtubule dissemble. This misfolding causes shrinkage of cerebral cortex as well as of hippocampus and enlargement of ventricles, etc. It results to memory loss, low learning ability, etc. Let's have a look on normal functioning of amyloid precursor protein and tau protein. The amyloid precursor protein in the neuronal cell membrane gets cleaved by the enzymes such as alpha secretase and gamma secretase in normal conditions. The alpha secretase enzyme cleaves amyloid precursor protein at N-terminal along with some part of beta protein segment. The gamma secretase enzyme cleaves amyloid precursor protein at C-terminal end. The cleaved segments are released outside the neuronal membrane. These cleaved segments are not in pure form, hence they are soluble. Amyloid beta protein in soluble form is non-hazardous. Hence, there is no accumulation of the beta amyloid protein. There are evidences that suggest amyloid precursor protein has some role in learning and memory, axonal outgrowth and restoration, and much more. But the actual function of the amyloid precursor protein in normal conditions is not yet been known appropriately. Thanks for watching.